Hello everyone, and thank you to the organizer for allowing me to report on the progress of the RTBfoods project, and in particular the work in progress on cassava. Many participants from the different RTBfoods partners have contributed to this presentation. I will highlight on behalf of the whole cassava team of the RTBfoods project the progress made on the development of evaluation method and protocols for the determination of quality traits of importance for breeding program, mainly related to processing or technological or functional behavior during intermediate preparation and cooking. The RTBfoods project, breeding RTB product for end-user preference, aim to pinpoint the quality trait that determine the adoption of new RTB varieties developed by breeders in 5 African countries: Benin, Cameroon, Côte d'Ivoire, Nigeria, and Uganda. The specific objective is to link local consumer preference with quantitative quality criteria before integrating these criteria into breeding program. The RTBfoods project analyzed 3 common use of each target crop: cassava, yam, sweet potato, plantain, and tropical potato. To establish specific product profile, the analysis used a reverse engineering approach, working backward from consumer to the breeders. RTBfoods focus on 5 work packages. Work package 1 look at socioeconomic aspect and identify the criteria that determine whether a variety is adopted or rejected. Work package 2 establish the link between the above user criteria and the biophysical properties of different variety. How does people preference, texture, consistency, taste relate to biophysical data, starch and fiber or pectin? Work package 3 built on the RTBfoods team expertise in terms of near-infrared spectroscopy analysis, NIRS and hyperspectral camera. These technologies serve to detect the biological components that make up food matrix. It is used as a fingerprint of foodstuff, providing information on certain quality traits, cooking quality, firmness, etc. Work package 4 investigates the genetic component of quality traits and assess the impact of the environment and their variability, such as climate and soil. Work package 5 assess whether the approach taken by RTBfoods has ensured that breeders increase their use of selection tools for quality traits and that new varieties have better match the user quality criteria. After the first 3 years of the project, this year was the opportunity to widely disseminate the first result obtained by the different teams for the characterization of quality traits, of great importance for user, processor, and consumer. A special issue of an International Journal of Food Science, International of Food Science and Technology, allowed the creation of a special issue dedicated to the achievement of the RTBfoods project. Despite the lockdown and the difficult situation faced by the project partner, all of them without exception participated in the special issue facing the comments of the reviewer. The special issue gathering 36 articles, 30 of them from RTBfoods team, is available in open access on the journal website thanks to the BMGF funding. 1 editorial, 5 review paper, 1 registered reports, which give an update of the methodology deployed by Work Package 1 to capture quality traits, and 11 original cassava articles, including 8 RTBfoods NextGen papers. The good news is that only after 6 months of publication, this special issue already gathered 58 citations as of today. Today in this presentation, we will focus on 3 cassava product profiles of great importance for the breeder of the RTBfoods project. Boiled cassava and its particular technological and organoleptic characteristics in terms of cooking in water, boiling or steaming. Gary has a ready-to-use semolina for making eba or drinks. Fufu with a particular focus on the aptitude for fermentation, the speed of softening of the roots, and the stability of the dough pieces during the conservation of the food. Great progress has been made in understanding the technological behavior of cassava pieces during cooking. Consumers agree that 20 to 30 minutes of cooking time is the maximum acceptable time for boiling or steaming cassava pieces. Many cassava genotypes are not suitable for cooking in water and produce hard, fibrous, or glassy pieces after more than an hour of cooking, which are not appreciated and are often rejected by consumers. The Cirad team, having a large American diversity of genotype for fresh consumption, with cooking time close to 20 minutes, has launched an ambitious crossing program to study the heredity of the traits and for the production of new genotype. In order to avoid the determination of cooking time with a fork to evaluate the hardness during cooking, different devices were set up to estimate the ease of cooking over time, in particular the speed of water absorption during cooking and the measurement of texture with a penetrometer or extrusion cell. Consumers prefer soft texture, which are very crumbly and break easily after cooking. The mealiness is a very important determinant of acceptability. You can see on the curve presented a great diversity of water absorption between genotypes, strongly correlated to cooking time and texture. This stat is now widely used by teams in Uganda and Colombia for population screening. This work was published under the DOE mentioned on the slide. All roots coming in the laboratory are scanned by NIRS on freshly grounded roots. Based on the first result of the 2020 harvest, the team working on the NIRS prediction after an appropriate mathematical treatment succeed in predicting two clusters, cooking time lower or higher than 30 minutes. 80% of the analyzed clones are grouped correctly in the corresponding class. The predictions are satisfactory and very promising to handle a larger number of trials performed on the present harvest. This could be a valuable tool for breeders working on the production of genotypes for cooking. The Benin and Uganda teams work on measuring the texture of clones after steaming, in parallel with testing of the same sample by the sensory panel. The standard of operation issued from the measurement of fairness was validated and are reliable for measuring root hardness at CIAT, UAC-FSA, and NACRE in Uganda. It was found that extrusion tests are the most repeatable and that penetrometric tests are the most suitable for the evaluation of friability or mealiness. By the evaluation of the hardness, a good correlation was demonstrated between the panelists and the instrumental measurement. It remained to determine with the panel the limit of acceptability of boiled cassava to define the acceptable limit force for the measurement of texture with texturometers. After validation of the SOPs, RTBfood team produced several datasets on the quality of their cassava population as shown in this table. Close to 800 measurements of water absorption and 600 measurements of texture were collected on more than 600 genotypes. Near spectra of fresh food were collected on nearly all these genotypes. The work package 3 NIRS team is developing prediction models based on texture and water absorption during cooking. A multivariate approach combining different reference data gives better prediction than trying to predict reference data separately. This tool may significantly accelerate varietal selection for cooking ability. For Gary, a lot of work has been done by IITA and NRCRI to develop protocols. The SOP for the production of Gary under laboratory conditions have been validated. The manufacturing of EBA has also been standardized and validated after numerous trials conducted on very diverse genotypes with different behavior from the NextGen program. Textural test was conducted and the SOP of EBA texture was validated at IITA. NRCRI used the extrusion test developed at Cirad for EBA texture evaluation. The sensory panel has been constituted and trained for the testing of the EBA at IITA and NRCRI. The correlation tests between the panel and the instrumental measurement are in progress. The IITA and RCRI laboratories have continued to develop NIRS protocol for measurement on fresh fruits. Better predictions are currently obtained for ground and homogenized pulp. Direct measurement on roots need to be improved to reach the same correlation. In the next period, a training session will be organized to standardize the SOP between the different partners of the project working on cassava with portable NIRS desktop and small device at IITA, NRCRI, CIAT, NACRI, and Cirad. A major effort will be put on protocol improvement, change in tool or methodology, applied, sampling representativeness, non-discriminant test method at Work Package 2 level. From the table, it is clear that NextGen clones were used for the development of the protocol with a reduced number of clones, 30 at IITA and 21 at NRCRI. Due to the COVID, the protocol could not be set up for population screening. These trials concerning instrumental and sensory tests, as in the case of boiled cassava, are essential to generate the NIRS database containing new quality data associated with spectral analysis. Prediction can only be made when the models are sufficiently robust. Fufu is obtained by soaking peeled or unpeeled root in barrel or pounds for 2 days. Many processed products are made from this type of fermentation. Fufu in Nigeria, lafun in Benin, couscous or baton as a stick in Cameroon, or shikwang in Congo. The performance of the fermentation is directly related to the productivity and quality of the processed product. Trials conducted at IITA on NextGen population show highly variable fermentation behavior. Fufu yield can vary from 5 to 30% of the initial roots. Nevertheless, a good correlation was found between dry matter and yield due to the mechanical grinding of the fiber. In most situations in Africa, fiber removal is done manually due to the lack of electricity, introducing a huge loss as can be seen in the attached photo. Fibrous variety versus non-fibrous variety for the same amount of initial roots. Only the central fiber of the root is removed from the best landrace variety used for rating on the right side of the picture. Fermentation duration impacts productivity, with soaking from 2 to 5 days and even more, and sun drying for 1 to 5 days and even more. Long fermentation impacts final fufu quality and preference due to the high acidity, bad flavor, and odor. Shorter fermentation produces soft fruit with an easy dewatering and fiber removal, with an increased surface for drying and the sun drying no more than 1 or 2 days, producing good final quality. Longer fermentation with hard roots, difficult dewatering, and fiber removal, with increased drying time, because there is no increased surface during the fermentation. The drying is between 3 to 5 days. It's mainly on this clone that aflatoxin appears during prolonged drying. The genotype of cassava used in soaking has a direct impact on food security. Woman drudgery increased for fiber removal. It's a manual process. Fiber removal time can be 2 to 3 times longer as measured in Cameroon. Different methods are being tested in the RTBfood project in Cameroon, Benin, and Nigeria to develop standard of operation that can be used to classify the different genotypes into 5 categories as very fast, fast, regular, slow, very slow fermentation. Experiments reported on the graph on the bottom left were conducted at NRCRI with very promising results for some NextGen variety, very fast genotype. The indicators are bubble and foam production, clarity of soaking water, speed of fruit softening. Another quality trait recently evaluated is the stability of the dough obtained during storage. Some lose their firmness very quickly after preparation of the dough, while others are very stable over time, as shown by the textural analysis presented in the slide and published at the DOE address. The dough losing their texture are very strongly rejected by the consumer. The functionality of processed RTB cannot be explained solely by fiber, starch, or amylose content. For boiled cassava, it has been shown that there is no relationship between these compounds, not even dry matter, and cooking behavior and cooking time or mealiness. The same pattern is observed for sweet potato. A team of the RTBfoods Project in Cirad, INRA, and James Houston Institute is conducting study on the role of pectin and in particular non-methoxylated pectin on cooking behavior. Pectin allows cell-cell adhesion and therefore have a key role during cooking. Preliminary tests show correlation between these pectic compounds and the texture of cooked product. Trials are underway and sample analysis in Montpellier on cassava from Cirad and soon from NRCRI and RITA. A flow analyzer has been purchased to conduct routine analysis for future NIRS prediction. Based on socioeconomic and technological study conducted with producer, processor, and consumer to identify the main quality trait and establish product profiles, many standards of operation are being developed. It was deliberately decided to work primarily on fresh fruits in order to search for compounds that can influence the quality of the final product. The screening of population must be of height and medium throughputs and proof of concept must be carried out for the development of rapid tests for detection and prediction of cooking and processing behavior. The various teams had made considerable progress toward the future integration of new quality traits into breeding pipeline. Ready-to-eat RTB products were rarely prepared and assessed prior to the release of new variety to evaluate consumer acceptability and possible varietal adoption by producer and processor. Additional training is still needed on NIRS, hyperspectral imaging, pectin analysis, ontology, and database management. But the generation of data on breeding program population should be initiated without further delay. We are very proud of the different team and work package who contribute together to development of this method. A big thank you to the stakeholders who put their product knowledge and local land race at the disposal of the project. Thank you very much for listening. Here are the RTBfood chefs. More information available on the RTBfood website. Thanks.